trials. Uh, just to just to give you an outline of what I'm going to talk about today, I'm going to first talk about the importance of study planning for small clinical trials. Particularly here, we're focusing on early phase clinical trials today. Talk about analytical approaches that have merit with clinical trials, but spend most of the time talking about several different designs that have been proposed for small clinical trials. I'll both talk about those from a more theoretical aspect, but as well as give some experience with designing trials for some early phase designs that we've had here at Iowa through involvement with NeuroNext. Just to do the standard notifications, this is being recorded. We have an open access mission with the course, and so this will be posted with the video and slides to the website. Will be publicly available. Just a reminder that you can use the chat function, as several people figured out, to let me know that you could hear me, which is much appreciated. And you can use the audience response when prompted. So just to start off and give some basic background overview, I thought it would be helpful to begin by just looking at this document that's now about a little over 10 years old, that came out as an executive summary for the National Academy of Sciences, looking at small clinical trials, issues, and challenges. In particular, I think it's helpful to look at the recommendations that came out of that document. So they had 6 main recommendations in the document: to define the research question, to tailor the design, to clarify methods when reporting trial results, to perform corroborative statistical analysis, to exercise caution in interpretation, and that more research on alternative designs is needed. So these are the recommendations that came out of this group for addressing small clinical trials, which was their focus. The interesting thing, I think, if you look at the recommendations, the items that are on this list, it's really no different from any other trials. So kind of the take-home message here, to kind of set the tone for the rest of the discussion, is that the need to have adequate study design, the need to have properly designed question with a study that's set up to answer the question, clear what your how you would know at the end of the study whether you successfully or unsuccessfully addressed the question or to move the science forward, is no different for a small trial, for an early phase trial, than for a late phase trial. It's more difficult with early phase trials, but the basic principles are no different. And in general, there's 3 basic requirements for any clinical trial. It should examine an important research question, it needs to use a rigorous methodology, and it needs to be based on ethical considerations to assure that risk to subjects are minimized. And again, that's true whether it's a large phase 3 confirmatory trial, a very early phase, phase 1, phase 2, wherever you are, no matter what terminology you use, this is all important. One of the things that's become increasingly clear as I've had a chance to work on designing other early phase and mid-phase studies is that it's much more difficult there, because the resources are limited, the size of these studies is limited, and there's often a desire to kind of do what looks like a phase 3 study that's just underpowered and smaller. And that, in some sense, doesn't really address anything. So if it's not powered to address a question, you get to the end of the day and you're going to go forward regardless of what you see, you haven't really clarified information other than you may have gotten parameter estimates or pieces of data that can be used to then design the future phase 3 study. But you haven't really addressed whether it's fruitful to do the phase 3 study or not. And that becomes a concern, particularly in a lot of neurological settings where the phase 3 studies may fail. And the phase 3 studies may fail because the phase 2 weren't rigorous enough, or the early phase studies were interpreted too positively. And that's an item that that needs to be focused on, to try to maximize resources by only taking things to phase 3 that have passed some rigorous methodology in the early learning phase aspects of the studies. So the problem with that is, as I alluded to before, with size limitations, resource limitations for these early phase studies, it's much more difficult to do this. And so the importance of adequate study planning is really magnified in small clinical trials. And that's a lot of what we'll focus on today. To give kind of an example, with late phase randomized clinical trials, you typically have large sample sizes, there's lots of standard designs, there's kind of lots of basic methodology to choose from. There are issues that you run into when you're addressing large trials. I don't want to underplay those. But in general, you can put up with a little bit of noise, you can put up with a little bit of sloppiness in the data, it will balance out in the end. Not optimal, but it'll balance out. So it's kind of like the first picture that you see here, where you're kind of running race around lots of different people. You know, if you make one bad move, maybe somebody will notice, but not everybody's gonna see what happened. With small populations, efficiency is critical. So you really have to squeeze every piece of efficiency out of every piece of information in the study. Use of models is helpful. You want to evaluate alternative designs. You want to really try to maximize the efficiency of the design because of the resource issues. And there, you know, it's like a run that you see at the bottom where you're kind of out on your own. So if you mess up, everybody that's watching is going to see because there's nobody else to take the attention away from you. It's kind of the difference between small and large trials. As far as designs of interest, I've got a list here. Obviously, there's not enough time to go into each of these in great detail today. However, I'll try to quickly touch on these, focus on some of the ones that come up more recently just to give you some basic background. The first thing I want to talk about is dose finding. So we're going very early in the learn phase here. We're in very early learning phase designs. Researchers may want to determine the maximum tolerated dose. So that would be the maximum dose that you could give to have some, that would achieve some threshold of toxicity. An accurate determination of that's important because what you're trying to do often, if the assumption is that as the dose increases, tolerability and efficacy would decrease. In order to have the maximum chance at having an efficacious dose, you want to take the dose that's as high as you can go without having safety concerns. So it's important to try to come up with a good guess of the maximum tolerated dose here in the early phase studies, because if you take too low of a dose, so you do a bad job and you're too low, you may take a potentially useful drug forward that might not show efficacy because you're not high enough in the dose frame. What's a bigger concern is if you go too high on the dose. So if you take too high of a dose forward, you might see safety concerns in future studies that would then hinder the drug development process. So you'd like to try to find at least a general sense of the target area to move forward into then your later learning phase studies and your confirming phase studies. The most well-known approach and one that's been used for a number of years, it came out of oncology settings, is the 3+3 method, where you basically start off at whatever lowest dose you envision. You treat 3 subjects at that dose, and based on the responses in those 3 subjects, you determine whether to keep moving up, to do more at the same dose, or to stop. So if you treat 3 and nobody has a DLT in those 3 subjects, you go up to the next dose level. If 2 or more of those 3 subjects have, you either decrease the dose or you would stop. Or if 1 had it, you would treat 3 additional subjects and see what happens to them. If you see another DLT, again, you would decrease and stop. If you don't, so you only had 1 out of 6, you would then go up to the next dose. You continue proceeding until you reach some stopping criteria or you get through your dose ranges, and then that's a way to select the maximum tolerated dose. This came out of oncology and has been used a lot in cancer centers. It's actually crept into neurology and been used in neurological settings. And some of the strengths of it is that it's a simple and intuitive algorithm. So, you know, it really doesn't require a high-level statistical input, a lot of design or operational expertise in order to implement. It requires no computer program and is relatively familiar to many clinicians. So it's pretty easy as far as designs go to implement. It's relatively intuitive once you understand the basics. But one of the challenges that comes into play, particularly in its use in several neurological settings, is that it's been criticized for treating lots of patients at low doses and not really producing a good estimate of the MTD. The reason that is when it's expanded outside of the range of areas where it's been optimized, it may not be as effective at finding an MTD. So it was optimized in oncology settings where the dose range that you're looking at is usually in the 30 to 35% range. If you're looking at lower dose ranges, so if your maximum tolerated dose is the dose such that no more than 10% of subjects have a DLT, then the 3+3 will most likely go much higher than you would intend to do. And you can see why by with just basic properties of a binomial distribution. So even if you're in the 30% range, so if the dose has, say, a 30% true toxicity, you've got about a 50% chance of stepping up based on the rules used in the 3+3. If you're down in the 10% range, you've got almost an 80 to 90%. So if you're trying to use a 3+3 design to determine a maximum tolerated dose for, say, a new agent for stroke, where your dose-limiting toxicity may be, major bleeding, and you don't want anything higher than 10%. If you use a 3+3, you're most likely going to select your maximum tolerated dose from the study as something much higher than the actual dose that corresponds to the 10% rate that you have, which obviously has negative consequences moving forward. To get around that, adaptive dose finding methods have been proposed. The most common and one that's used probably the most is the continual reassessment method. There are a number of papers about the method. I think one of the best for a simple tutorial is one that appeared in Statistics in Medicine almost 10 years ago now, which is a very easy to read tutorial which gives the basics of the method in a non-mathematical, non-statistical for the most part assessment. So this is slightly, it's got the same basic objective that the 3+3 has, but it's set up in a slightly different way. So you start off with some assumed model, which can be very crude to select the doses, and then you treat cohorts. So you may still treat 3 subjects or 1 subject or 4 subjects, however many you want in the cohort, and determine whether they have a DLT or not. After that, you then refit the model. So as you get more information, the model is going to converge to what should be the true dose response curve, and you choose the selection of the next dose for the next cohort based on the curve, which incorporates both your prior information and the information that you've observed by the subjects as they go through the study. To implement that, as I mentioned, you go through this process, you refit, and you continue until some stopping criteria are met. The stopping criteria could be some maximum number of subjects have been treated. You treat some fixed number of subjects at a single dose, in which case you might stop and say that's the maximum tolerated dose, or other stopping rules that have been suggested, or if the dose response curve changes by some tiny threshold, you would then stop and say that it's converged and use that to select your maximum tolerated dose. I think it's helpful visually to see how it works. So take the following example, which is a very simplified example, which assumes you have continuous dosing and you're going to treat 1 subject in each cohort, just to illustrate the purpose. So you start off, let's say you start off with the following curve, and your target toxicity level is 10%. So you would trace over the 10% occurs at dose 5. So you would start at dose 5. That's your dose that's closest to your defined dose toxicity threshold of 10%. And you would treat the first subject at that dose. When you proceed on, then that first subject is the purple dot that you see here. That does not have, they do not have a dose-limiting toxicity, so you refit the curve, which is the purple line that is higher. So you would then shift the dose toxicity curve down, that which would correspond to the blue curve. The blue curve then shows a dose of 12. You would treat the next subject at a dose of 12. The blue dot indicates that they had a dose-limiting toxicity. You would refit the curve, and then eventually you would iterate. As you can see, it starts to move less, so that as you get to the end, once you've converged and stopped, your final estimate for the maximum tolerated dose is the dose that corresponds to 10% on your fitted curve, which corresponds to somewhere around dose 7 or dose 8. Now, there are some advantages of this approach. It learns from information that's obtained, so it doesn't just use the data from an individual cohort. It uses the data from that cohort and every cohort that has been observed up to that time point. It's generally more efficient than the 3+3 design. There have been lots of publications that have addressed this point, that have shown that you can more accurately estimate the maximum tolerated dose with the CRM. You're less likely to treat subjects at doses that are far from the MTD, and you're going to treat more subjects at the MTD or at doses near the MTD, which has advantages. So you're going to get more precise estimates in the area that you're most interested in, which is around the area where the MTD happens. And you're also less likely to treat participants at ineffective doses or at toxic doses. So it achieves kind of several objectives. There are drawbacks to the CRM, the biggest being you have this statistical model that has to be refit using Bayesian methods, so there needs to be a much more substantial collaboration between investigator and a statistician. Any clinician with a basic understanding of the 3+3 can run one of those with really minimal involvement of a statistician. With the CRM, you really need a statistician to be involved in the study team and more involved in the day-to-day activities of the study. The properties really need to be assessed by a simulation that can be done. That holds for the 3+3 too, but it's more of an issue with the CRM. A bigger concern that was raised with the CRM, and kind of the example that I went through illustrates it, is there's a safety concern, and that early on you may have very limited data, and that those first cohorts are going to swing the curve a lot. So for example, in the situation that I just went through a few minutes ago, we started at a dose of 5. Since we didn't see a dose-limiting toxicity in that first subject, the curve was shifted quite a bit, and the next estimate based on the curve for the dose at 10% would be 12. So if you stuck strictly to the CRM based on that curve, you would more than double the dose in your next subject. There are concerns that have been raised, rightfully so, that that might be too much and might cause safety concerns. Because of that, what's normally used now is more of a modified version of the original CRM, where you would always start at the lowest dose under consideration, regardless of what your prior information may tell you. You would always enroll, say, 2 to 3 patients in a cohort, and you would restrict any dose escalation to no more than one level or some defined rule that you would have in place. So in a lot of ways, with the modified CRM, what those effectively do in a number of situations is you start off with what looks basically like a 3+3 design. And then once you see your first toxicity, then you implement the modeling approach and it kind of takes over. And the CRM component really drives your future cohorts more than the fixed step-ups that you have in the 3+3 design. So that's early phase. We're now going to shift a little bit to look at a number of different designs that can be used in early phase studies, but more later in the learn phase. Some of these are the same type of designs that you would see in later phase studies. I mention them because some of the aspects of them are actually a bit more important to consider here. So for instance, repeated measures design. So these would be designs where you're measuring some outcome over time. And often we might have baseline measurement, a 3-month, a 6-month, a 9-month, a 12-month. Now, these can be helpful because those repeated measurements give you information. What's often done is when we analyze that, we might analyze change over time. So maybe we get measurements at baseline 3 months, 6 months, 9 months, and 1 year, but our primary outcome is the change over 12 months. And so we would use the 12-month value minus the baseline value and analyze the change. Perhaps we compare the mean change in the treatment group and a placebo group to see whether there is more or less change in the treatment group. A better approach would be to take that final value with the baseline value as a covariate. That's typically done in large trials. What's not done as often, and again, this is a situation where you can get away with it in larger confirmatory studies, but it may be more critical to address in these early phase studies, is really modeling the longitudinal nature. So using a longitudinal model to take into account the correlation of the measurements in the same subject over time. By doing this, if you can adequately model that correlation, you can reduce the variance of the comparisons that you're most interested in, which squeezes you a little bit of power out of that, which again, remember in these early phase studies, every bit of efficiency is critically important to squeeze out. So if you're collecting data at baseline 3 months, 6 months, 9 months, 1 year, you would like to incorporate all of that data in the analysis, even if you're primarily interested in the 12-month outcome, because of the efficiency gains that you can get from looking at that longitudinal data. Another design that comes up a lot is crossover designs. These can be beneficial. I mean, crossover designs are kind of a simplified case of a longitudinal design, except you're changing the treatment at each time. So with a crossover design, rather than randomizing half of the subjects to treatment and half the subjects to placebo, each subject will get both, and what you're randomizing them to is the order. So maybe half the subjects get the active treatment first, then there's a washout period and they would get the placebo, whereas the other half of the subjects would get the placebo first. After a washout period, they would then get the active treatment. It gives you the advantage of each subject serves as their own control. Again, you're able to take account of the correlation of multiple measurements within subject to reduce your variability. But there are some disadvantages. So you need to have a long-term disease where relevant outcomes can occur and be measured during the time period you're talking about. And you also have to be careful that there's no carryover effect. So carryover designs work well when if you stop treatment, someone then goes back to the rate they were before treatment. If they're better, then it's going to make a difference which way you're given. So if I treat someone first, and then I look at their placebo, but if there's some carryover benefit from the fact they were treated, they're not the same as a person who got placebo before they were treated. And it's going to cause differences when you then compare the two groups because of this carryover. That's one of the most challenging aspects of designing crossover studies, because it's really more of a hypothetical argument. It's very hard to prove carryover. It's kind of like if you're trying to prove that all swans are white. If I find a black swan, I've disproven that. But the fact that I've never seen a black swan doesn't prove that all swans are white. I would have to see every swan in the world. It's kind of the same issue here. We're often underpowered to assess carryover effect for the methods that can do it. So it's very hard to rule that out, but the success of it depends on that. There are disease areas where a strong case could be made for that, and it can be very useful, but it's something that has to be carefully considered. when that is being employed. One of the things, particularly for learn phase, that's become of more interest in recent years is a special case of crossover design called an N of 1 design. So this is very useful, for instance, for an extremely rare disease where it'd be hard to have a sufficient number of subjects to do a study. You can use an N of 1 is basically one individual enrolls in the study, and they have successive period of time where they're treated or on placebo or on treated versus control treatment. And so each subject receives the experimental treatment for one time period of each pair and the control for another time of each pair. And they may have multiple pairs of treatment periods in which they rotate from one to the other. And within each pair, the order of the treatment is randomized much in the same way that you would randomize groups of subjects in the crossover design. That then gives you the chance to look at that, and you could then take into account the correlation among an individual subjects, look at how that subject has done when they were treated versus when they're on placebo, and make determinations based on how the time periods, the multiple time periods for that subject compare. You could also do N of 1 studies for multiple subjects and then combine them using meta-analysis technology, and there's been a few publications that have used that. It's a little tricky to do. It takes for the right situation, again, you got to worry about crossover effects, same as you do in the crossover design. But for instance, one of the areas where I've seen it recommended is like for asthma, if you're looking at asthma exacerbations. So somebody who's asthmatic, look at it like a maintenance type of asthma medication. How many asthma attacks do they have when they're on maintenance versus when they're off maintenance? Because you would expect those periods to be relatively comparable. And you could do that over maybe your pairs are 2 months. So 1 month you're on, 1 month you're off, and you do that over a course of a year, and you get several time periods where a subject is on treatment versus the time when a subject is off treatment, and you could compare the asthma attacks during both those time periods to get a sense of how effective the treatment is. Other types of designs, there's selection designs, which these are very, very simple designs. These are really for a situation where you may have a disease for which there are few or no treatments that exist, and you might have multiple candidates. And you would like to study the candidates, but you don't have the resources to kind of do each at a time, and you really don't have a clear sense of which one might be have the highest promise to look at over time. So selection design, it's essentially a horse race. You could do a very small study where maybe you're looking at 4 or 5 different treatments, and you allow them, so you treat different groups of subjects at each of the treatments, and whichever one responds the best would be selected as the winner and would then move forward to get more scrutiny. The selection design is not intended to be a rigorous hypothesis test. It's intended to try to pick the best candidate out of a set. If you think about it from a type 1 error, it's got a type 1 error of 50%. So even if none of the drugs work, you're going to pick a drug to go forward based on the way that you've set it up. The future studies would then assess whether there's benefit of that. But you're trying to look at, I've got 5 candidates, I'm going to put these into a study, realizing I don't have power to look for efficacy for each one in this study, but I would like to get evidence to see which one shows the most promise to focus in on that I could then use my additional resources on. So it's kind of a nice screening tool in kind of the early to mid phase learn aspect. One that's actually, I think, received more focus, and we've designed a few of these in NeuroNext, is a futility design. It's kind of a poor choice of names because people, I think, get scared by the phrase futility. But what this, you could also think of this as a non-superiority design. So many of you are familiar with non-inferiority designs where you're trying to show that some new treatment is no worse than an existing drug. This is kind of flipping that in the other direction, where what you're trying to do here is say, is there enough evidence here in this smaller study to determine that a treatment's not going to be effective? Or, I can't say that it's not effective. I can't say it's effective, but it looks promising enough that a phase 3 might be of interest. And so what we do to set this up is you need to specify what's the clinically meaningful effect of interest, which this might be the effect that you would power a future phase 3 study on. So if you say that a 10% increase in a favorable response rate is clinically meaningful, then a futility design would set up with a null hypothesis that the treatment improves the outcome by at least that 10%. And your alternative would be that it does not improve the outcome by at least 10%. So with this, it's different from your typical null that we would see in studies where the null is that the 2 groups are equal versus an alternative that they're different. Here, if you reject the null hypothesis, You're saying that the study has shown that there's enough evidence that I can reject the null and claim that the treatment doesn't improve outcome by at least 10%, which I've said was my minimum threshold. So it would be futile to proceed on to a phase 3 study. Whereas if I don't reject the null hypothesis, I have not ruled out that I wouldn't see that 10% effect. I haven't shown that there's a 10% effect because I haven't powered it to where I'm really trying to address that there's 10%, but I'm saying that there would at least be enough there to suggest moving forward. This would also give you preliminary data to power that phase 3 study. You would have more data on your control event rates, your other aspects of the study that will be important for that, but it achieves the question, or it achieves the primary objective that I alluded to early on, that it does so in such a way that you have a question that you've set up and addressed in the study. You can see here just the difference in the hypothesis. This just reiterates what I mentioned, how the hypothesis for the futility and the usual design is very different. The type 1 and type 2 errors are different here. From the usual design, a type 1 error is when we determine that an ineffective therapy is effective, and a type 2 error is when we determine that an effective therapy is ineffective. Those are flipped in the futility design, right? Because we would make a type 1 error if we say an effective therapy is ineffective. So we declare futility when we actually have a treatment that works, and we'd like to make that very small. The type 2 error is if we say an ineffective therapy is effective. So something that should be futile does not get declared futile and proceeds for a phase 3. Right? That's going to have resource issues because then we might have a phase 3 study that's negative, so we'd like to minimize that. Whereas the other has potential financial implications if it's a biotech or a company or someone who is interested in the development of the treatment under interest. With futility designs, the way that they're typically set up, negative predictive values are high, and that's to address those concerns with the type 1, type 2 errors. So with most futility designs, if you declare futility, the probability that the treatment is not effective is pretty high. But the positive predictive values are not that high because you haven't powered the study to show efficacy. So if you, the probability that it's effective, given it's not futile, is not necessarily a given. So not declaring futility is not the same as showing efficacy. It just suggests that it should be looked at further in a phase 3 study. So it's kind of, if you think of it, it's kind of provides a benchmark that you have to cross this benchmark in order to say there seems to be enough evidence to look forward, which really seems to be an adequate way, kind of as an intermediate step to address this concern, to have rigor in your learn phase while not requiring them to be the size of a phase 3 study, which would be much too complex to conduct at that point. The last thing that I want to quickly touch on here is adaptive designs, which some of these can be used, the designs we talked about can be used as an adaptive design, or adaptive designs are really much broader than that. So just to give some background, many of you have heard of adaptive designs. One of the things that I think causes a lot of confusion with adaptive designs is that they've been around for a number of years now. They've received a lot of discussion in the literature and in studies that have started to use adaptive designs. But it's a little unclear what exactly we mean by the phrase adaptive designs. There are very many different types of adaptive designs. A group of individuals could be talking about adaptive designs they're working on, and those designs could have very little in common. There's just that many different designs that could be used. So it helps to have like a uniform definition. And luckily, in recent years, two definitions have come out, two separate definitions that have a lot of similarities in terms of what constitutes a valid adaptive design. The first came from a pharma working group on adaptive designs, which is now under DIA, as a DIA scientific group. But when that group was formed, in 2006, they published a white paper, which was the first time that a formal definition of adaptive design that kind of appeared in the literature. Their definition has a couple of important aspects to it. So they define an adaptive design as a clinical study design that uses accumulating data to modify aspects of the study as it continues. And then there are three important points after that. Without undermining the validity and integrity of the trial, the changes are made by design, not on an ad hoc basis, and it's not a remedy for inadequate planning. So by this definition, an adaptive design is one in which you have limited information going into your study. There are aspects that you would like to know that you don't know, and you would like to start your study, get some of that information, and then, given what you've learned, make the final decisions on those aspects of your study. If you can pre-specify that, the reason that's important is you can simulate it. You can run simulations under a range of situations, both with what you expect it to happen, other situations that may be counter to what you expected to happen, to look at issues of bias, type 1 error, other negative consequences that could come in from the adaptation, and make a case that your adaptive design is satisfying the operational characteristics, that you are not undermining, in their language, the validity and integrity of the trial. In 2010, the FDA released a draft guidance document on adaptive designs where they also included a definition for adaptive design. And their definition was very similar to that used by the Adaptive Design Working Group, a study that includes a prospectively planned opportunity for modification of one or more specified aspects of the study design based on analysis of data from subjects in the study. So both of these definitions really focus on this phrase adaptive by design. So in some sense, adaptive design is a misnomer and it has a poor choice of name. It's not adaptive in the sense of what the word adaptive means, which you kind of respond to changes on the fly, right? Species adapt because something unexpected happens external to them and they have to make modifications in order to survive. That's really not what we're talking about here. A better term would probably be pre-planned adaptation. So you going in, you specify up front what you don't know, what you might do. So if A occurs, I will do this. If B occurs, I will do that. If C occurs, I'll do this. And then once the data occurs, it's then kind of laid out what you would do once you have the data, as opposed to I'm going to start, get the information, and then we'll figure it out once we have the information. That's really not under either definition of an adaptive design. This figure, this comes from a paper that was recently published and really just serves no purpose other than to illustrate that there's an infinite number of adaptive design possibilities. Just shows some that have been proposed. There are many, many more than this that could be proposed. There's kind of an infinite number of possible adaptations that could be considered and used in various studies, both early phase and late phase. One important thing that comes out of that definition with adaptive by design is something that's been commonly used in clinical trials. Group sequential designs for interim monitoring meet that definition. So they have pre-planned adaptations based on existing study data, where here the adaptations are relatively simple. It's either stop the study early for efficacy, stop the study early for futility, or continue the study. And most, if not all, large clinical trials, even a lot of mid-phase trials now use these types of techniques. One of the important things that I think is often not appreciated by researchers is when you take a course in clinical trials, you may cover these methods and you'll talk about things like Pocock spending bounds, O'Brien-Fleming spending bounds, the Landamet spending function, and you learn how to incorporate those. And it's actually pretty standard in clinical trials. What's really not appreciated is the implications of using those in a smaller sample setting. So all of these techniques are based on large sample theory. If you open like a basic clinical trials textbook, it'll often have a phrase, something like, for simplicity, we're going to assume the variance is known, and then those procedures are illustrated. That's a very simplistic way of saying in statistics what we mean is we're assuming that the sample size is large enough so that the estimate of variance we have will converge to the true variance. That means that the properties of these techniques are based on what we call in statistics asymptotic theory or large sample theory. If your sample size is large enough, these methods work well. If your sample size is small, these methods may not guarantee control of the type 1 error. So if you're in a very small, maybe 100 subject study and you're doing an interim analysis after 25 or 30 subjects have been done, you use O'Brien-Fleming, which is pretty general that you've used and been taught to use in clinical trials, you may not control the type 1 error because it's a large sample property that you have. And I see there's a question about the reference from the last trials. The slides will be posted, which will include the reference, or we can be sure to send it after this. Another thing that's been proposed, adaptive seamless design. So these are designs that try to combine different adaptive features. In some ways, what this tries to do is the big selling point of these is to reduce the white space. So in general, you proceed from one phase to another phase. You finish one, you look at the data, and you use that in order to plan the next phase. But there's some downtime or white space during the period of time that you're in between the two. Whereas if you know what you need to know from the first phase to go into the second phase, you could do a seamless design where you go straight from one into the other once you have that information. An adaptive design does that and uses data from subjects in both in the final analysis. So it can be very efficient if set up in the right way. Most of the literature on adaptive seamless design is focused more in the confirmatory phase, where you're going from a late phase 2 into a phase 3. Maybe you have several doses of treatment. You use the early phase of your seamless to choose a dose, and then you go into a phase 3. Use all the data to do your confirmatory study at the end. I'm not going to focus on that here, but the reason I mention this is that there have been instances that have crept up in recent years of using these adaptive seamless designs in early phase studies. And I'll just mention one example of that is a study in ALS that was done out of Columbia University, where they did an adaptive seamless design, which was very interesting, that looked at two of the different designs we've talked about. So they did a selection design as the first phase of this adaptive study, where they looked at two different doses of coenzyme Q10 versus placebo, with the objective being that the selection design would be used to choose one of the two doses. That would then go forward into stage 2, and in stage 2, they would then collect additional subjects in placebo and the selected dose of coenzyme Q10 for a futility design. And then at the end of the study, a test would be done for the futility type hypothesis that we discussed with the futility design, comparing the subjects treated on the selected dose of CoQ both in stage 1 and stage 2 with the placebo subjects in stage 1 and stage 2. And based on that, a decision would be made of whether it was futile to continue to phase 3, or whether this suggested there was enough evidence that a phase 3 study should be considered with coenzyme Q10 in ALS. So these have the potential. They have a lot of attractive features. So your question, the endpoint used to select dose was the ALS functional rating scale, the change in that over time. I think it was from baseline to 12 or 24 weeks. So adaptive seamless designs have potential to improve the drug development process. They have a lot of appeal in reducing this white space. It does require specialized methods, because you're now using data-driven methods. So you use data to make a decision, and then you're using that same data at the end. So for instance, going back to the example I mentioned, you chose the dose in the first stage that was best. If neither dose was better, you're still incorporating randomness that's going to make that dose look better in stage 2. So there's a positive bias that you're carrying forward that often needs to be addressed at the analysis phase. It could be done, it just takes time and care to do that. And so the benefits of compressing the white space may be offset by there's additional analysis challenges that need to be addressed either by a simulation or in the design aspect. Just something to keep in the back of the mind. It's not problematic, but it's something that needs to be recognized. One of the things that I think is critically important with adaptive designs, and this is where a lot of confusion comes into play, it's important to be clear what an adaptive design can and cannot do. This comes up a lot in the rare disease setting, or in settings where there aren't many treatments that exist. And what I've seen there is individuals involved in those disease areas or research areas often hear adaptive designs, and they see this as something that's great. This will help get treatments to this field. And the adaptive design itself is not going to do that. It's a tool. It's not going to make an effective treatment ineffective. It's not going to make an ineffective treatment effective. It's only going to give you tools to better answer the question. One of the selling points of adaptive designs that's often discussed in talks is that it can allow you to more efficiently, get drugs to market, to show that a drug is effective. And that can be true in situations, but in my experience, the biggest benefit of the adaptive design is not on that side, it's really on the other side. And what adaptive designs can really do, which is also useful, is to more efficiently identify when there are treatments that aren't effective, or when you should not look at a treatment any further, which is disappointing from the study team side when you're interested in trying to see that treatment. You're disappointed it doesn't work. The disease community is disappointed. But as a scientific community, that's still an advantage, because you're then not using additional resources for a treatment that may go on to prove ineffective. You can then take your resources and start to invest them in other areas which may prove to be more fruitful. And so if adaptive designs can be used more, particularly in the learning phase studies, to better identify ineffective treatments so that those never get to phase 3, that's an advantage for the clinical trials community, because failing at phase 3 is much more problematic, much more expensive, much more costly than failing at phase 2 and identifying that a treatment is ineffective at phase 2. One of the things with adaptive designs is there's a lot of, so I see there's a question, can I compare adaptive designs to group sequential design? A group sequential design is an adaptive design based on the definition of pre-planned sample size. So The way I see it in the figure, let me go back just to clarify that question. If you look under adaptive designs, I would argue that group sequential designs fall under adaptive designs. It's a subset of adaptive designs. Adaptive designs could go beyond that, but a group sequential design and interim monitoring is a very special case. It can be combined with others, so you might do interim monitoring with sample size re-estimation, which is adding complexity, but it in and of itself would fit the definition, both from the FDA and from the adaptive design working group as an adaptive design. So going back to the adaptive designs, one of the things that has come out from the adaptive designs is there's a much greater need for infrastructure building if you're going to do these adaptive designs, because some of the issues that we've talked about require fairly extensive simulation studies in order to assess the properties, to make sure that you're controlling the integrity, the validity of the study, and to make a case that the adaptations that you're implementing are not going to have negative consequences for the study as a whole. That takes a lot of upfront work. So adaptive designs are thought to be more efficient, reduce time, but they require more upfront work on the team. That's typically a challenge from an NIH setting if you're working on grants that are funded through NIH, because a lot of the time that might be required to do the simulations, to do the justification, is before you can even write and submit the grant. And so there's often not funding to support the time of the clinicians, the study team, the statisticians that need to have these discussions and put this together. And it's an area where, to be frank, academic trials are behind industry trials, because a lot of companies have now built teams to do this type of modeling and assessing of designs, and that doesn't exist as much in an academic setting. So developing that infrastructure on the academic trial side is needed, and there have been some recent steps towards doing that. One that I mentioned before that we're involved with here at Iowa is NeuroNext, or the Network of Excellence in Neuroscience Clinical Trials, funded by NINDS, which provides infrastructure to help conduct early phase trials in the neurosciences. One of the advantages of this, because of that infrastructure, if an investigator comes into NeuroNext with a problem that might fit an adaptive design, there is infrastructure funding to help pay for the time of the clinicians and the statisticians who would work with the investigator to help do these upfront things, to justify the design, to make sure that we've selected the best design, to compare the adaptive design to a fixed design. It might be that the adaptive design has some theoretical promise, but when you actually run the comparisons, you gain very little from a fixed design, in which case, implementing the complexity of an adaptive design may not be worthwhile. So just to summarize, this was a lot of information, I recognize. I tried to give an overview of a lot of different types of early phase designs. I'll go back to the point I raised at the very beginning, that an appropriate study design, whether it's in a late phase study or an early phase study, has a sufficient sample size, adequate power, proper control of bias to allow a meaningful interpretation of the results, or state another way, that it can answer the question of interest. And there are limitations, there are challenges. Designing early phase trials is, I think, much more difficult than designing late phase trials from the statistical aspects, from the clinical aspects, because you're limited by what you can do often. You need to come up with a question, you need to come up with a way to answer the question, but you can't just keep adding sample size, or else the size of your study gets too large. So one of the objectives of this course is to provide the students who have applied and would go into the study teams to bring ideas for studies that they come in, and then to help them get the tools, or to see how to approach that, how to pull teams together that can address this. And so with that, I will stop. Just as a reminder, there is a website which you see here with a link, which I'll leave up for a few minutes. And again, this will be posted, that you could go to evaluate the webinar. I see that there is a question to comment on the use of a control group and blinding in first-in-human phase 1 trials, with the design intention being to obtain some early evidence of efficacy. I've seen that come in. There are two sides to that, and it's one of those where it depends on the specific study. There are advantages of having like the control group, having the blinding to try to get some early evidence of efficiency, which can help to inform the future, the next studies, and to help identify how best to approach. The drawback to that is it often complicates the study. And it really depends on you've got competing goals here. One is to answer the main question of interest in your first-in-human study, to try to find which dose looks most promising based on safety or whatever you're looking at. And then the other is to try to see whether there's a hint of efficacy, which might be important from a more global standpoint of what to do next. Those are kind of competing with each other. If you put in a control group, you put in blinding, you now complicate the safety component of the study, because now you've got to maintain that blinding, you've got to figure out how that's going to be implemented. So I don't think there's anything wrong with doing that. I think that it has to be recognized that it's going to implement challenge. It's going to make it harder to address the question you initially set out to address, but it's going to give you more information at the end. So it's something that has to be weighed by the study team and everyone involved to see whether the added complexity is worth the extra benefit that you get out of it. And I think that has to be addressed on a study-specific basis. So this is what I have. I guess we have about 10 minutes left where I can take any additional questions or clarify anything that was unclear. Before we do that, I'd like to thank everyone that joined today. It's nice to on a Friday, everybody's looking forward to the weekend, to join. I appreciate it. All right. Well, if nothing else, we'll end. Again, the slides will be posted. My information will be available if anyone has questions or something that they want to follow up on. I'd be glad to try to answer any follow-up questions via email. So with that, again, thanks for your time, and hopefully you'll join future webinars. So there'll be an additional series of these, both before the course, after the course, and the intent of these is that these webinars can be a tool not just for people participating in the course, but for individuals who would like to know more about aspects of clinical trials, particularly in neurology. So if you liked it, spread the word for future webinars, and we'd like to see more people. Thanks a lot.